Okay, so it was originally thought that the heart is a post-mitotic organ, in that the cardiomyocytes you're born with, you also die with. This has now changed due to the identification of cardiac stem cells, which are resident in the adult mammalian heart, and these have a role to play in contributing to the myocardial mass by actually putting back cardiomyocytes that are lost as a result of wear and tear, aging and injury. Now, the first identification of new cardiomyocyte formation was found in the human heart in 2001, published in the New England Journal of Medicine by Antonio Beltrami. And what they found were these small mitotic myocytes, which you can see here on the left. And these mitotic proliferating myocytes were in the border zone of an infarct. So this showed for the first time that the human heart was actually having some kind of proliferative effect following an infarction. Then in 2009, this paper was published in Science, where they showed actually that there is myocyte turnover in the normal adult heart. And what they did is they did carbon-14 dating, and they showed that the cardiomyocytes are replenished over the course of the lifetime of a human, but this is a very, very small amount. So actually it means that your heart is replaced, probably about 50% of your heart is replaced throughout your whole of your lifetime. There are other investigators that say that this is actually increased, and the actual times that your heart replenishes itself over your lifetime is maybe 4 times. So there is a high controversy about the actual turnover and replication of the cardiomyocytes in the adult heart. So the fact that we have new cardiomyocytes forming in the heart, albeit a very low turnover, the idea was that where could these new cardiomyocytes actually be coming from? And it was this paper that was published in 1995 that postulated towards that these cycling cells could actually be myocyte progenitors that derived from a more primitive cell and therefore still immature and not yet irreversibly withdrawn from the cell cycle. So this was the first indication in our community that there could actually be stem cells in the heart. And then from the same group, in 2003 came the identification of adult cardiac stem cells in the adult mammalian heart, and these were found in the rat heart. These particular cells, and these are the ones that we work on in our group, express the stem cell marker c-kip, which you can see here on the surface of the nuclei in green. They also express other markers which are reflective of the origin of the heart. So for example, NKX2.5, which is a marker of cardiomyocyte, or is expressed in cardiomyocytes that are newly developing. So these cells, which are c-kip positive, have properties of stem cells in that they are clonogenic, multipotent, self-renewing, and they have significant regenerative potential. If you take the middle row here, you can see that a c-kip positive cardiac stem cell can progress into an adult cardiomyocyte. It starts to upregulate transcription factors which are specific for the cardiomyocyte lineage, such as GATA4 and MEF2C. It can then undergo a few rounds of proliferation, shown here by BRDU labelling, before it becomes a terminally differentiated cardiomyocyte. So these cells were identified in the rat myocardium. We also needed to ascertain if they were present in the human myocardium. So we set about to find these cells, and here you see 2 c-kip positive cells in the human myocardium. We were able to obtain some samples from each of the 4 chambers so that we could look at the myocardial distribution of these cells, and we found that they were most prevalent in the left atria, or the atria, and also the left ventricle appendix. And this is not too dissimilar to what we find in our rat and mouse models. So can we isolate them? Yeah. So we can take a small piece of human myocardium, and this is a human atrial sample, and then we're able to isolate them in 2 ways. We can either do an enzymatic digest, where we digest the tissue with enzymes so that we can get a small cell preparation, or we can do a thing called explants tissue culture, where we cut the atrial sample up into tiny, tiny pieces, and then what we do is we plate these pieces, we put them into an incubator, and then cells migrate out from these small pieces, and then we're able to select those cells. And then what we do is we use a process called magnetic activated cell sorting, where we have our antibody, which is attached to a magnet, and then we pass our cells, which are then positive with this magnet connection, the antibody, and then we're able to get a pure population of our c-kip positive cardiac stem progenitor cells. So the amount of c-kip positive cardiac stem progenitor cells that we find in an average atrial biopsy is about 4%. So 4% of the small cells that we've obtained, 4% of them are c-kip positive cardiac stem cells. So what happens with the effects of aging, also gender, and pathology? So what we found is that the number of cells that we can isolate from a given sample doesn't change depending on the age of the patient, and doesn't change depending on the gender of the patient, and also doesn't change on the pathology. So we can obtain approximately 45,000 cardiac stem cells per gram of tissue, and this work is done in collaboration with the surgeons at Liverpool Heart and Chest Hospital. So what we have to do once we get our cells is we have to prove that they, even though they express c-kit, there are other cells in the heart that also express c-kit, namely being cardiac mast cells. So we have to show that our cells are the actual stem cells. So we put them through tests. The first test is clonogenicity. So we have to prove that a single c-kit positive cell can make a clone, or can basically derive other cells that come from that single cell. And we show that our human c-kit positive cells that have been derived from all 4 chambers have a similar clonogenicity to what we find in the mouse and the rat, being about 30%. We then check them for their profile to see that they express stem cell markers. And as you see here, one particular clone expresses c-kit and expresses pluripotency markers OCT4, NANOG, BMI and TERT. It's negative for the endothelial progenitor marker CD34 and the hematopoietic marker CD45. And we confirm this by immunostaining. As you see here, they're c-kit positive in green, and they express pluripotency markers OCT4, NANOG, BMI, and they also express the cardiac transcription factor marker, which is reflective of their origin, NKX2.5, and a small amount of them express islet-1. So we also perform an assay to prove that they're multipotent and that they can make all 3 cardiac cells. So we put them through a process where where we make things called cardiospheres, which are basically balls of hundreds of cells, and then these cardiospheres are plated, and then cells in the periphery spontaneously differentiate into the 3 main cardiac lineages, being cardiomyocyte, shown here in red, and smooth muscle actin in green, and von Willebrand factor, which is the endothelial marker in white. However, this assay only allows us to detect cells that are very immature, in that they've differentiated into an immature phenotype. They don't show any kind of sarcomeric structure or beating in this assay. So what we wanted to do is we wanted to develop a cocktail, and so that we could make our cells differentiate into functional cardiomyocytes. And what we did is we took information from our colleagues that work on embryonic and iPS cells and how they differentiate them in vitro into beating cardiomyocytes. So what we did is we took our cardiospheres, we plated them in a specific medium, and we showed that over 14 days our cells started to express the sarcomeric actinin and started to show sarcomeric structure and ZEG disc formation. They also started to express connexin 43, showing gap junction proteins between them. They had downregulation of their stemness markers and upregulation of their cardiac specific markers. And they did beat, but I don't know if it's going to work. No. Sorry about that. Okay, so we had gone from a very primitive cardiac stem cell to a beating cardiomyocyte in the dish. So the final assay that we have to do to prove that our cells are indeed the stem cells is whether they have in vivo regenerative potential. So we take some of our cells and we transfect them with a fluorescent protein, here it's green fluorescence, and then we inject our cells into the border zone of an infarct. and we inject 500,000 of these cells at 5 different sites in the peri-infarct area. And then 28 days later, we look for new cardiomyocyte and new vasculature formation. So here you see we have plenty of green cells, so we show that our cells are within the infarcted area. When we double stain for cardiac troponin I, which is cardiomyocyte marker, you see that some of these green cells are also cardiomyocytes. And also you see these structures in the bottom left corner, which also look very much like vasculature, new vasculature that's been formed that is also green. So we show that our cells have contributed to new myocardium. So what happens with the function? So we looked briefly at the changes in the function, and we show that when we inject our human cardiac stem cells into the nude rat, we show that we can actually improve the function, but obviously it hasn't returned completely to normal. So, as you know, every cardiologist's dream is to mend a broken heart, and stem cell therapy is becoming a very kind of significant area that people are focusing on to try to do this. There are a number of stem cells that people are using and looking at. The bone marrow, which is already in the clinical arena, and also the skeletal muscle myoblasts, and then obviously the ones that we've identified in the heart themselves. Now, the first autologous cardiac stem cell clinical trials have taken place, and these were published last year in the Lancet. And what they did in the top trial was they took 500 to a million cardiac stem cells, and they injected them they did a phase 1 trial into patients with ischemic cardiomyopathy, and this was called the CPO trial. And then the other trial is taking a different set of cardiac stem cells called cardiosphere-derived cells, and they basically did the same thing, but they're just a different cell type. So what were the results? So they showed that at 4 months and 12 months after the transplantation of these cells, there was an improvement in ejection fraction, and there was also a change in the infarct size in the CPO trial. In the Caduceus trial, they showed that there was also a decrease in the scar size, but they didn't find a change in ejection fraction in this trial. And the main difference between the two trials is one used MRI and one used echo to measure the changes in ejection fraction. So these trials have predominantly been thought of as phase 1 trials which have shown an effect, an improvement, but actually it's been, because they injected such a small amount of cells, probably these cells are not actually giving rise to new cardiomyocytes. What they're actually doing is having a paracrine effect. And this paracrine effect is what is most explanation which is given for the bone marrow trials as well, which is that you're injecting these cells which are then going to the heart, and they're having a kind of stimulatory effect and also a cell survival effect on the cells that are at risk, and also they're inducing angiogenesis, and then they're also stimulating the resident cardiac stem cells so that they can maybe go on to do some kind of job. So that's where my research primarily comes in, and what we're looking to do is we're looking to do regeneration without actual cell transplantation. So we recently published this paper last year in the European Heart Journal where we looked at the effects of exercise on cardiac regeneration. And what we did is we took some rats and we randomly assigned them to different groups. So we had a control group, we had a low intensity exercise group, and we had a high intensity exercise group, and they exercised to their individual VO2 max. They did this for 4 days a week for up to 4 weeks for 30 minutes a day. And we found that this exercise regime was enough to induce cardiac stem cell proliferation and therefore an increase in their number. And this also led to new cardiomyocyte formation, and this amounted to about 7%, so it was quite high. This new cardiomyocyte formation was then reflective in an increase in cardiomyocyte number with the 2 intensities of exercise. and it led to favourable changes in the function of the animals. So we're also part of an initiative which is funded by the EU FP7 programme, where we're looking to devise a therapy which is affordable, readily and widely available and easy to apply and compatible with the current clinical standard of care. And this is called a CareMI. And in CareMI we're looking for specific therapies where we can activate the endogenous cardiac stem cells. So I'm going to talk to you about one therapy, which is our growth factor therapy. So the problem that we have as basic scientists and also translational scientists is that we use a lot of our models on mouse and rat. But when it comes to the heart, we really do need to use a model which is similar in size to the human. And that's because a mouse left ventricle weighs 250 milligrams and is less than 1 millimetre thick, whereas a human left ventricle, which you can see here, weighs about 400 grams and is 1 to 2 centimetres thick. So the amount of myocardium that we're trying to replace in a mouse MI is nothing in compared to what we're trying to replace in a human. So we need to use a model which is more similar to the human, so we chose the pig model. So the pig heart, because it is similar in physiology and anatomy to the human heart, we wanted to test 2 growth factors which we know to have an effect on cardiac stem cell biology, which is IGF-1 and HGF. And we injected these growth factors into coronary after MI in the pig heart, and we looked at the effects of these growth factors on survival and regeneration. So the pigs were given an infarction through occluding the left anterior descending coronary, and then they were reperfused for 60 minutes, and then 30 minutes after the reperfusion we injected saline as our control, and then we injected 3 doses of the growth factors IGF-1 and HGF. We implanted a BRDU pump so that we could measure cardiac regeneration, and then we sacrificed them up to 21 days for histology and 2 months for function. So we showed that our c-kip positive cardiac stem cells, they express the receptors for the growth factors, so which means that they will become activated as a result of the growth factors being there. The number of c-kip positive cardiac stem cells significantly increased, and this increased linear to the to the dose that we gave. They proliferated, and also they differentiated into the 3 lineages, which are the NKX 2.5, which shows their commitment to a cardiomyocyte, GATA6 and ETS1, which shows their commitment to the vasculature. We also measured the myocyte regeneration and new myocyte formation, shown here by BRDU labelling. And we found a significant increase in the number of BRDU positive myocytes, and again this increased linearly with the dose of growth factor that we gave. And we also found new vasculature, as you can see here, and new capillary formation of the growth factors injected. Finally, the function. So we did MRI at 2 months. We confirmed our regeneration results at 2 months. So what we'd seen at 21 days was sustained up to 2 months. And what we did is we found that although the function wasn't necessarily improved, it had not worsened. So it had been sustained with the growth factors that we delivered. So in conclusion, the adult mammalian heart, including the human, harbours tissue-specific stem progenitor cells, which are clonogenic, self-renewing, multipotent, and exhibit significant myocardial regenerative capacity in vivo. We found that aging, gender, and pathology does not affect the number of these cells, and they can be officially isolated from human myocardial samples. Autologous stem cell therapy necessitates patient-specific tissue harvesting and cell processing, with delays to therapy and possible variations in cell potency, and it's going to be expensive. We've shown that exercise training induces physiological cardiac growth encompassing stimulation of the endogenous regeneration mechanisms. And growth factor stimulation therapy is an off-the-shelf, safe and effective cardiac regeneration therapy which results in improved myocardial remodelling, function, and stimulation of endogenous cardiac stem cells and the result of myocardial regeneration. We believe that the future of cardiovascular regenerative medicine is dependent on our success in dissecting the biology and the mechanisms that regulate the endogenous cardiac stem cell fate, and this information will provide the tools for the design of the most physiologically relevant clinical regeneration protocols. I'd like to thank the people of my laboratory and also my collaborators in Italy and in Paris, and my mentor Bernardo Mzal-Genard, and also our collaborators at the Liverpool Heart and Chest Hospital and the funding bodies. And finally, thank you for your attention.